Hello, everyone, and welcome to today's webinar, Immunodeficient Mouse Models to Support Prolonged Engraftment of Human NK and Tumor Cells. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Envigo. To learn more, visit envigo.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Jenna Frame, PhD, Senior Manager, Scientific Communications and Marketing, Biocytogen, Boston. Jenna, you may now begin your presentation.
Great. Thank you so much, Kaylee, and to the team at LabRoots for hosting today, and to Envigo for the invitation to present to you all. So today's presentation will start with an introduction to the BNDG model and its general utility. I'll then outline the applications of the BNDG mice and second-generation models that were further engineered based on specific needs for testing the role of the human immune system in testing novel immunotherapies. These will include specific case studies highlighting their capabilities. So for BNDG mice, this includes testing anti-tumor therapies in CDX or PDX models. For the B2MKO strain, the possibility of extended studies involving PBMC-derived T cells. And finally, the ability of the BNDG humanized IL-15 mice to better support engraftment and survival of human NK cells for NK-based immunotherapies. Of course, then we'll be able to answer any questions you may have. So there are a few different versions of immunodeficient mice out there. Our BNDG immunodeficient model, which was generated by Biocytogen, hence the B, was generated on the NOD-SCID background, which is significant to mention because it has some predilection for being more receptive of human cells due to known differences in the murine SERPA allele. And similar to other strains in the market, these mice have a null allele for PRKDC, which is involved in VDJ recombination, and they also have mutations in IL-2 receptor gamma, which is obviously a key cytokine receptor critical for development of functional lymphoid and NK cells. So owing to these additional mutations, compared to the NOD-SCID predecessors, these mice have a longer lifespan and minimal incidence of thymic lymphoma because they have essentially no lymphocyte leakage, which you can see on the right-hand side here from the representative data measured by flow cytometry, with essentially no B, T, or NK cells present as shown on the top right. Furthermore, there is no measurable presence of any IgG or IgM in the serum of these mice measured by ELISA, shown by the black bars on the bottom right. So together, these characteristics allow for efficient engraftment of human cell line-derived or patient-derived xenografts. So there are several applications that these mice can be used for when engrafted with human cells. These include tumor and tumor stem cell research, which I will focus on today, as well as research involving embryonic stem cell and iPS cell engraftment, differentiation, or functional potential. Similarly, basic hematopoiesis and immunology studies are often carried out in these mice to discover the alterations in hematopoietic or leukemic cell potential or transformation potential, as well as in the context of infectious disease. And finally, these models can be further engineered by introducing more human genes, for example, to support a specific human cell population of interest. And at Biocytogen, we're continually introducing new specialized humanized models for these exact purposes. So since our work at Biocytogen and Envigo is mainly focused on preclinical immuno-oncology studies, I will present applications of these models in the context of these types of analyses. So here on the right side is a simple proof of concept study demonstrating higher engraftment of Raji B lymphoma cells into BNDG mice, which is demonstrated by the shorter survival curve, which is shown by the red line in the panel A, and the drop in body weight in panel B, and significantly higher expansion of the lymphoma tissue in the BNDG mice compared to the non-skid or the nude mice counterparts shown in panel C. And on the bottom is the evidence of tumor spread, both grossly as well as by histology in the liver and spleen of these mice, as seen in part E. So when we consider the types of human cells that we want to engraft for testing novel anti-tumor therapies, we can employ a couple of different strategies depending on the type of therapeutic that we're interested in examining. So if we're directly targeting tumor cells, we can simply engraft those into the mice and test the efficacy of the candidate therapeutic. This could start with a cell line-derived xenograft and move on to individual patient-derived xenograft models. And if we're interested in understanding the interplay between the human immune system and the human tumor cells, there are a few different strategies we can employ outlined on the right side. So if we're interested in the role of T cells or purified NK cells, for example, PBMCs can be engrafted directly or further purified. And I'll introduce an example a little bit later in the presentation where we purified NK cells to do just that. And if the goal is to harness the role of and or the interplay of multiple immune cell types, then the engraftment of CD34 positive HSCs would be an ideal choice. So here's a sample efficacy study in a CDX model where the target antibody is directly targeting tumor cells. So in this case, Raji lymphoma cells that can be tracked by luminescence activity. So in this study, the antibody was given either at day 10 post-tumor cell injection or on day 3 and day 10 with this dual treatment group of mice shown on the bottom row in panel A. So as you can see from the representative images and by the blue curve on the right-hand side, the animals receiving both treatments on day 3 and day 10 had significantly slower tumor growth than those receiving a late treatment or no treatment at all. In this next example, injection of CAR T cells targeting human CD19, which is expressed on the same Raji lymphoma cells, were injected into BNDG mice 2 weeks after the lymphoma cells were transplanted. And on the right-hand side, you can see images of the control and the CAR T infused mice over time, which demonstrates a substantial reduction in tumor growth resulting from the CAR T cell infusion, while the untreated mice, which is shown on the left-hand side, were unable to survive past 3 weeks. These CDX studies in BNDG mice are also amenable to multiple different experimental arms. And in this case, 4 different versions of CAR T cells shown on the left-hand side in comparison to the black vehicle-controlled mice. So as you can see, each experimental group showed substantially different tumor growth kinetics owing to the differential efficacy of these particular CAR T cell populations. And on the right-hand side, you can see the body weight of the mice with the highest tumor burden starts to drop near the end of the study. So in addition to human tumor cell lines, BNDG mice are also excellent recipients for human patient-derived xenografts, all known as PDX models. Here is a summary of all the different types of PDX models that have been established, with their success rates varying depending on the tumor type. Notably, though, altogether, over 300 different PDX models have been successfully established in BNDG mice by in vivo. Here's an example proof of concept study demonstrating the successful engraftment of a triple negative breast cancer xenograft into BNDG mice with the tumor growth kinetics shown by the black line. So the treatment of these tumor-bearing mice with kinase inhibitors, which is shown by the red line, was very effective in blunting the tumor growth compared with the control group, which proves that the PDX model in this case can provide a platform to efficiently evaluate the in vivo efficacy of anti-cancer drugs. So in these next couple of slides, I'd like to introduce how BNDG mice are effective recipients for human immune cells and hematopoietic stem cells. So in this particular example, human PBMCs were injected into the mice and reconstitution was evaluated by flow cytometry. As you can see on the bottom row, with PBMC engraftment, the cell type reconstituted is largely T cells, with both CD4 and CD8 T cells expanding in these mice. However, as shown by the reduction in body weight and survival, many of the mice start to die after 5 to 6 weeks due to graft versus host disease resulting from the large numbers of T cells engrafted. Now, despite these characteristics, PBMC engraftment of these mice can still be extremely useful for testing T-cell immunotherapies that may not need a long experimental window for treatment. In addition to PBMCs, CD34 positive stem cells can also be engrafted into BNDG mice. Now, the cons of this experimental setup is obviously the longer experimental time window for engraftment, and as well as the radiation of the mice, which is why the uninjected mice, which is shown in panel A by the red line, fail to survive. However, the advantage is the multilineage reconstitution of these mice. And as you can see here, there's a high percentage of CD19 positive B cells as well as CD3 positive T cells. And this reconstitution can be maintained for several weeks post-transplantation. And in this study, you can see at least 16 weeks. So here is some example efficacy studies for CD3-based bispecific antibodies in BNDG mice reconstituted, in this case, with human PBMCs. So on the left-hand side, the BNDG mice were engrafted with human PBMCs and then inoculated with NUGC4 gastric adenocarcinoma cells. These mice were then treated with a bispecific antibody targeting CD3 and Claudin 18.2, which showed excellent efficacy in this model at the higher doses, shown by the green bars. Similarly, on the right-hand side, treatment with blinatumomab, the CD3-CD19 bispecific antibody, is very successful in this humanized PBMC model at preventing the growth of the Raji lymphoma cells, as shown by the luminescence data, again, on the bottom row. So to provide a brief recap of the relative advantages and disadvantages of PBMC versus CD34 positive HSC reconstitution in immunodeficient mice, this chart, I think, provides a very nice summary. So the human PBMC engraftment is more of a transiently humanized model with overall lower levels of CD45 positive cells engrafting, with 20 to 30% of the total CD45 population listed here in the chart. And this occurs rapidly over a period of about 2 weeks. However, due to the majority T lineage engraftment, GVHD begins to occur in the mice relatively quickly, meaning that the planned studies will need to be more short-term and generally targeted toward T cells. On the other hand, the CD34 positive engraftment model is a more stable humanized model that requires more time to establish, but results in multilineage reconstitution that is helpful for assessing agents which require multilineage immunity or assessing characteristics like memory responses. However, what if you're working on targeting human T cells and need an extended therapeutic window to examine efficacy? So this need in the field led to the engineering of a new second generation model for this exact purpose. And this involves specifically negating the MHC1 response, which on the left side and left panel you can see is responsible for presenting endogenous antigens to CD8 T cells to license cell killing. So in order to negate this response, the beta-2 microglobulin region of MHC was targeted, which is shown on the right side with the green part of the protein. However, because the beta-2 microglobulin protein is a subunit for both MHC as well as FcRn, which is responsible for binding and recycling of IgG, we needed to get a little creative with the targeting strategy. So together, this led to the following targeting strategy in these mice to attempt to reduce the onset of graft versus host disease in these mice. So disruption of the endogenous beta-2 microglobulin allele is shown on the top right, with the addition of that same cDNA into the FcRn locus, so as not to disrupt the function of FcRn. Thus, we have a new BNDG model with MHC1 essentially disabled. So this prevents T cells in the graft from attacking the host cells, but allows for normal presence of beta-2 microglobulin for IgG recycling. So here shows the experimental validation demonstrating the lack of MHC1 expression in the BNDG B2MKO mice, shown on the top row here, when compared to the parental BNDG strain and the non-skid mice, indicating successful generation of the mouse model. We also confirmed that MHC class 2 expression in dendritic cells was intact, as you can see on the right side. In this case, the dendritic cells were gated by CD11B and CD11C expression. And we can see that MHC2 expression is unaffected compared again with the parental strain in the not-skid mice. Next, we wanted to confirm that the normal function of FcRn and IgG recycling can be maintained because the beta-2 microglobulin gene is now under control of the FcRn promoter. So as you can see in this graph by the red line, the concentration of human IgG in these mice after injection is relatively comparable in the KO mice compared with the parental strain and the C57 black 6 mice. But it is not detectable in another B2MKO strain that does not rescue the beta-2M expression in the context of IgG recycling. And so that's why this strain is also named B2MKO+. So we next tested the engraftment capacity of PBMCs in these mice using 3 separate donors in each column here. And as you can see by the blue lines, the BNDG B2MKO+ mice were able to survive significantly longer than the parental strain when engrafted with the human PBMCs from each donor, with a significantly lower incidence of GVHD symptoms, as shown on the bottom row, and with each donor, which includes signs such as lethargy, lack of response to stimulation, hair loss, and so forth. So together, these data allow us to definitively conclude that the BNDG B2MKO mice have delayed onset and reduced severity of GVHD compared to the BNDG parental strain. Here's another sample study with a little bit longer timeline, just so you can appreciate the extended time window that this model offers. So flow cytometry data shows comparable engraftment of leukocytes and T cells from 2 separate donors, with the green and the orange from the same donor, and the red and the black engrafted with cells from the second donor. So the BNDG mice have reduced survival and exhibit fluctuations in body weight, shown by the black and green lines on the bottom, while the B2MKO mice are able to survive past 100 days and exhibit no reductions in weight, which would be one indicator of GVHD that is monitored in these mice. So together, these data indicate that there is indeed an extended PBMC engraftment window in these mice. Next, we wanted to test the anti-tumor efficacy in these mice. So in this study, the combination of pembrolizumab or anti-PD-1 and ipilimumab or anti-CTLA-4 was assessed against RKO cells, which is a poorly differentiated colon carcinoma cell line, which was engrafted into the BNDG B2MKO mice plus. So as you can see by the green line on the top left, the treatment was very effective compared to the untreated mice, in which the tumors grew much faster. And on the bottom row, we've stratified the individual tumor growth kinetics to further demonstrate the clear difference between the treatment groups. Now, the average body weight of the mice, as shown on the top left, in contrast, remained comparable between the groups, suggesting that there was no adverse effects of the treatment on the health of the animals. So the results indicate that this BNDG B2M mice plus, reconstituted with PBMCs, does provide a powerful preclinical model for in vivo assessment of therapeutics targeting human T cells for anti-tumor therapies. So to recap the benefits of the BNDG B2MKO model, the engraftment of PBMCs is tolerated by this model much better, owing to the loss of recognition of mouse antigens by T cells via MHC1. This significantly extends the treatment window by several weeks to allow for more robust experimental results. What about models to test immunotherapies that involve other cell types? So targeting NK cells to promote tumor cell killing is a recent area of interest that is showing a lot of promise, as is the generation of CAR NKs for next generation of cell-based immunotherapies. So one key cytokine regulating NK cell development is IL-15, which regulates the activation and proliferation of the cell population. So in order to provide a model that provides support for NK cell survival, we generated a humanized IL-15 model that can be used for engraftment of HSCs, PBMCs, as well as NK cells directly. So in this model, the IL-15 cDNA was inserted into the mouse IL-15 gene after the 5' UTR. Expression of human IL-15 in this model was confirmed both by mRNA expression on the left-hand side, where the mice were also confirmed to lack murine IL-15 expression. Similarly, expression of human IL-15 was detected from mice stimulated with poly-IC, and this was not detected in wild-type mice. Together, these data indicate successful generation of the humanized IL-15 model. So in addition to confirming expression of human IL-15, we also performed some proof of concept studies to show that the humanized IL-15 strain better supports human NK cell engraftment and maintenance. So in this study, the PBMC-derived NKs were pre-sorted and engrafted into either BNDG humanized IL-15 mice, shown by the red dots, or the parental BNDG strain, shown by the blue dots. So as shown, NK cells were able to better persist over time in the humanized IL-15 mice, measured both by counts on the top left and the proportion of NK cells in the CD45 population, shown on the right. In addition, as you can see in the chart on the bottom, on the far right, NKT cells do engraft and persist in this model as well. Further phenotypic analysis of the NK cell CD56 positive populations confirm elevated expression of functional NK cell markers and receptors in the BNDG strain containing humanized IL-15, such as granzyme B, NKG2A, NKG2D, and NKP46 receptors. And other functional NK markers such as perforin, CD107, NKP30, and CD57 exhibited similar trends. So while PBMCs or NK reconstitution in these mice can be a useful tool for NK and T cell based experiments, as reconstitution is often biased toward T cells with PBMCs engraftment, CD34 positive engraftment can also provide other immune cell types that may be of interest. So as you can see from the reconstitution data in this example, engraftment of CD34 positive cells can also support engraftment of B cells, modest levels of myeloid cells that can persist for months after injection. Thus, there are no deficiencies apparent in the multilineage reconstitution in these humanized IL-15 mice, and they maintain obviously a significant boost in the NK cell reconstitution as expected, as you can see on the top right. In this next series of experiments, we show establishment of several CDX models in the BNDG humanized IL-15 mice engrafted with human CD34 positive cells. So after engraftment, mice were grouped and implanted with 3 different cell lines depicted on the right-hand side. After this, the numbers of leukocytes, T cells, and NK cells were assessed weekly over a period of about 5 weeks. So as you can see from the red bars, in contrast to the black bars, the humanized IL-15 mice showed significantly higher levels of leukocytes, T cells, as well as NK cells after tumor cell engraftment. So these data suggest that the capacity of the engrafted immune cells is indeed improved due to the presence of the human IL-15 gene in these mice. In this next case study, BNDG humanized IL-15 mice reconstituted with CD34 positive cells for 6 weeks were subcutaneously injected with human A549 adenocarcinoma cells that were genetically engineered to express human claudin 18.2, which is a popular tumor-associated antigen. So after 7 days, mice were treated with the human anti-claudin 18.2 analog antibody regimen. And this group is shown by the red curves on the tumor growth scale, which showed efficacy in slowing tumor growth compared to the IgG control, which is shown by the black line. So on the right-hand side, you can see that CD56 positive NK cells and CD3 positive T cells were present both in the blood, as shown in the middle panel, as well as the tumor, as shown on the right-hand side, extracted from these mice at the endpoint. And together, this sort of indicates that this model is very powerful for evaluating the role of both NK cells as well as T cells in potentially slowing tumor growth in combination with other anti-tumor treatments. So to summarize the utility of this model, insertion of the human IL-15 gene really allows higher numbers of functional NK cells to persist in this model after engraftment and reconstitution of CD34 positive human hematopoietic stem cells. And ultimately, this yields a more efficient model for testing NK-based immunotherapies. So to summarize everything that I've presented today, BNDG mice are extremely valuable preclinical research tools used to assess the functional potential of a variety of human cell types for a variety of applications. So they can be engrafted with multiple human tumor cell lines and primary tumors for efficacy studies in BNDG mice. And for immuno-oncology studies, BNDG mice can also be engrafted with human immune cell types to investigate the role of the human immune system. And in particular, these second-generation models are extraordinarily useful. So the BMC engrafted BNDG beta-2 microglobulin KO mice allow for extended treatment windows due to the lack of MHC1 recognition by T cells. And the CD34 positive engraftment of the BNDG humanized IL-15 mice is an ideal choice for evaluating the NK-based therapeutics due to the enhanced survival and persistence of human NK cells. So Thank you so much for joining us today, and I hope we have enough time to address all the questions. If you have any further questions after the presentation, there's some contact information here. You can contact us at Biocytogen or our colleagues at Invigo using the information on the slide. And I'd like to invite Kaylee back to start the Q&A session.
Awesome. Thank you, Jenna, again for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. So let's get started. We've got a few questions already in the queue here. So we'll start with this one. It asks, what is the difference between the NSG and NOG strains and BNDG mice?
Yeah, that's a lot of alphabet soup. But in general, I think the strains are very similar. So the BNDG strain is on the NOD/SCID background, not obese diabetic, and further modifications were made to the IL-2 receptor gamma gene. And so I think the strains might differ in the amount, the type of genetic editing, but essentially the same genes are disrupted in these mice.
Thank you. Next question here. Are there special housing conditions required for maintenance of these strains?
Yeah, so the BNDG mice are actually maintained in normal isolator conditions. We recommend that they receive acidified water to prevent bacterial growth, but generally they can live quite a long time in those conditions.
Great. Another question here. Are there any other BNDG strains that are going to be available?
Yeah, so there are several different BNDG strains that Biocytogen offers. And, it really depends on the type of study that you're considering and the type of cell types, human cell types that you want to engraft and flourish in these mice. And so we've tried to take all those considerations in mind when we're generating new models. And so if you're looking for a specific model, definitely reach out.
Wonderful. All right. Well, we will go ahead and wrap up there. I want to thank you again, Jenna, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Invigo, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you had provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.